there are many other millions of species doing fascinating things. And so this is how I like to think about the pragmatic value of our study of nature.
Gracias. ¿Alguien más quiere comentar? ¿Alguien más, por favor? Eva, por favor. Y Marco.
Hola, bueno, antes que nada quiero agradecer a todos ustedes por aceptar la invitación de mis compañeros. Es la verdad un gusto y un honor que hayan podido venir. Y bueno, la pregunta que les quiero hacer es más bien como una preocupación personal. El cambio, bueno, el uso de las herramientas con las que nosotros podemos estudiar estos patrones y los procesos que observamos cambia muy rápido, ¿no? Primero veíamos RPLPs, ahora secuencias, microsatélites, ahora Next Generation. En su experiencia, ¿como cuánto tiempo ustedes creen que el uso de los microsatélites pueda todavía estar presente? Porque, bueno, esto es algo que, bueno, yo en lo personal estoy trabajando ahorita con estos datos, pero ya hay otras herramientas y hay que estar al día, ¿no? Hay que dominarlas para poder seguir en esto.
I'm going to translate. Brian might be very interested in this question. She's saying that, commenting and asking that, given this extreme revolution of methodology, not only analytical, but using from AVLPs to sequences to next generation, if we believe microsatellites will come to its their day, or if there's hope for us that we use microsatellites. That's my interpretation.
Well, I can certainly give you my opinion about that. I think microsatellites have a mutation rate that makes them extremely useful on a level that even a densely sequenced SNP panel might not be useful on. And so, you know, I think the really exciting thing about new sequencing technologies is that you can now directly tailor the sort of data you're collecting to the types of questions that you have. And so if you're interested in phylogenetic questions like Frank, maybe you go after these anchored or ultra-conserved elements. And if you're more at an intermediate level, maybe, you know, rad sequencing or something, the real fine-scale landscape genetics, I think most people would argue that microsatellites provide a really good source of data for that. So I certainly don't see them going away. What I do see is it's now easier to discover microsatellites. And for instance, students in my lab have done things with anomalous regions using a relatively long read sequencing, and then you can mine those regions for microsatellites, and all of a sudden you have 100 prime pairs and you can multiplex them and make the data collection more efficient.
I'm not sure I understood the question, but I'm getting, supposing that if the question involved microsatellite loci and their utility. Yes. We use microsatellites a lot in my laboratory, but for different purposes. For the purposes of assessing genetic parentage, which is really genealogy or phylogeny across a single generation. We can do maternity and paternity analyses. And when you accumulate such information for multiple families, then you can deduce the mating structure, the mating system of a species. So I think microsatellites they're like enzymes on steroids. They're just highly polymorphic nuclear markers with nice Mendelian properties. In the context of genetic parentage analysis, they certainly have some utility also in polyography, but not so direct, I would say. It's going back to more traditional kinds of population genetics where you're dealing with allele frequencies and looking at structure from that kind of a perspective. What has really surprised me a lot about the field of phylogeography is that 10 years ago or 20 years ago, I would have predicted that the new revolution would be in getting nuclear gene sequences analogous to those provided by where you could, as sequencing technologies got better, I was imagining that one would be able to go into the nuclear genome and pluck out pieces of DNA, say 20 kilobases long, and do genealogical analyses analogous to those provided by mitochondrial DNA. If one could do that, that was what I was imagining might happen in the next 20 years, then one could have nuclear gene trees be directly comparable to one another and to mitochondrial DNA. The technologies have not enabled that to happen in quite the way I envisioned. Instead, we've gotten sort of almost, I would say, sidetracked by some of these other technologies like microsatellites and more recently, RAD sequencing and the SNPs that they provide. So I would hesitate to predict what the future will bring because I've been so wrong about the past.
Sí, quedó contestada tu pregunta, Eva. Sí, ¿verdad? ¿Quién seguía? Marco, por favor.
Bueno, well, following just a little bit the Pepe's question, I want to know if you're concerned about this kind of disparity that you show, for example, in your lecture. Your data set, your analysis just blew our minds, especially in Mexico. I think in the charity record, we don't have this kind of data sets and technology for do a kind of competition or complete all these data sets. I want to know if you're concerned about this disparity or imbalance that new technologies like next generation cluster analysis, stuff like that, has caused.
Bottom line, it sucks. I think it's terrible. I mean, I want everybody to be on the same page. You guys have world's diversity. I mean, that's one aspect. You have the world's diversity, and right now we have capability. So with my Brazilian collaborators, they send their students up to me, and they bring their samples. We extract them, and we send them to those places and get the data generated, and then they learn how to analyze them either in my workshops or with me. And I think, unfortunately, I clearly can't control any system in Mexico or Brazil on, generating these things or how long it will take. I mean, it's a symptom of money, right? Of having to have these money and infrastructure. Anybody else have any?
I have a very good answer.
Oh, good. Okay.
So you asked your question in English, sorry. And this partly pairs on the previous question about microsatellites. I think as scientists, we often have this sort of addiction to the very latest, best, most expensive, fanciest technology. And we don't necessarily always need that level of power. The choice about the markers that we use or the genotyping techniques that we use really needs to be driven by the types of questions that we want to answer. And for some types of questions, in fact, so I did not present this, but some of the work that we do in my lab is simply CRRPs because it's cheap and faster and it works. So in some ways, I think one can still be answering interesting questions without using the latest technology. However, there may be questions that you seek to answer that do require genome scale data. The identifying the variants associated with style length in yuccas was something that requires genome scale data. And my answer to that is that I think that it is eminently possible for people who are working outside the US or in smaller institutions to be able to do that type of work. Something that may not be clear in my introduction is the college I work at is actually very small. We serve only undergraduates. I have the only postdoc on the entire campus. I'm the only one. But we've been able to do this genome level sequencing in part by library development or sequencing. And we've also been able to make use of a lot of computational infrastructure that's available in the cloud. So we don't have a supercomputer at my institution, but I can rent time from Amazon. And so I'm able to do a lot of those same, not purely to the quality that Brian does, but some of the same kinds of things without having major infrastructure at my institution. It's just a matter of building appropriate collaborations and finding ways to do it without having to build an entire sequencing facility. Does that make sense? No, I don't know, maybe you are, but...
Yo también quisiera agregar en español un comentario y enfatizar que realmente depende de la pregunta. Regresando entre la pregunta que haces ahora y la pregunta que hizo Eva, depende realmente de la pregunta y el tiempo que necesita uno para contestar esa pregunta. Si uno está interesado, por ejemplo, en una pregunta que tiene que ver con la relación de parentesco entre individuos en una población, pues a lo mejor lo que es la respuesta, como ya lo explicó John. Pero si son preguntas a nivel global sobre patrones, por ejemplo, bibliografía ecológica, a mí me parece que lo que tenemos que hacer en el futuro es desarrollar capacidades no solamente para el acceso a las tecnologías y los costos que eso implica, sino también nuestras capacidades de análisis y de colaboración. Incluso, creo que la presentación de Frank es obvia en ese sentido, de que para poder abordar estas grandes preguntas a nivel continental, también se necesita una gran capacidad de colaboración. Y entonces eso evidentemente multiplica la complejidad de los recursos que se necesitan no solamente para colaborar, sino para poder obtener resultados.
I had one more comment really quickly about that. Well, I do believe that you can
answer. I do believe that you can answer questions with a few number of loci. Always, and I've had this happen. The possibility that you're wrong because you have a few number of loci, and that's a really creepy feeling, right? You feel like you're rolling dice and hoping that it is. You can always be wrong. You can always be wrong, but you want to, like, if you have the ability to do it, you want to eliminate the parts that are likely to be wrong, right?
You should quantify your
uncertainty. Exactly. Right. So I probably don't need 300 or 400 loci to generate this tree. As Brian said, you probably need 50. But for me now, it's easy to get 300 as it is to get 50. Okay, so, but for my questions, I want the trees to be as best as they can for the downstream comparative approaches. And if those are messed up, then the downstream parts of that suffer. And they probably still are messed up, right? There's always things you can't take into account, but you don't want it to be
based on one or two loci where there is a probably higher probability of that being wrong. So while you can try to answer them with that, there is always that unfun feeling that there is something going on. So try to get as many loci as you can. You know, I don't know what to say about that.
I think the other thing is that it's really clear to me that these costs are going down precipitously and very fast. And when I first heard about next-gen sequencing, I was a postdoc at the University of Michigan. It was probably 2006. And they were talking about costs for doing just a really modest amount of sort of a proto-red sequencing, so like $50,000 or $60,000 for the level of phylogeographic study. And my postdoc advisor, Lacey Knowles, you know, said there's no way that we're doing this. Brian could just do a lot of PCR in the lab. It's, you know, and then when I got to LSU a few years later, and we started doing our own RAD-seq. This was on a 4x4 sequencer. The sequencing costs were much higher. We probably paid, you know, between $20,000 and $25,000 US for one study's worth of data. And at the time, it was a lot of data, and now it's a laughably small amount for that sort of study. The RAD-seq that we're doing now for a comparable level of sampling is 100 times as much data, and it's costing us between $2,000 and $4,000. So this is just in, you know, less than a decade that the costs have decreased, you know, very rapidly. And I think with any new technology, what happens is the technology will dissipate from the field. More and more people will be able to use it. It gets applied to more and more different types of questions. And that's encouraging, I think, for all of us, because we can do more science and we can get better answers as these technologies decrease in price.
So what I'm wondering is, you may have you may have these costs coming down in the U.S. where you already have the facilities and the infrastructures that are running, you know, thousands of these machines. But I don't understand. I don't know how it works, like in Brazil or here, how you build that infrastructure, those facilities to have the, you know, somebody running 100 Illuminas and doing it and processing. So while you can get the system cheaper just because of all chemicals and everything goes down in price, you still have to have the facilities to do that. I don't know, are they in existence here? Or?
It's across the river. It's across the river.
It is.
There are a few in Mexico, like one here, one in La Regia, in another state. But that's my very personal opinion. I think it is still cheaper for us to have these amplifications and the whole laboratory thing done outside Mexico than here or in Argentina. But I'm going to switch to Spanish. Y quiero volver un poco a las dos preguntas, pero sobre todo a la segunda pregunta de Marco, donde él decía, bueno, si tenemos preocupación de esta disparidad entre lo que sucede en el trabajo que se puede hacer en el Norteamérica respecto a lo que vamos a hacer en México y más al sur. Y ya se han mencionado muchas cosas, hay que pensar la pregunta, hay que tener la visión de colaboraciones sin duda. Y yo añadiría que sobre todo ustedes están a punto de empezar un proyecto, qué van a hacer el postdoc, o en fin, para iniciar su trabajo, y les interesa la geografía, la biogeografía, y están pensando en algún grupo de especies cuya distribución sea compartida, así como puede ser compartida de Centroamérica hacia el sur, como lo que yo les mostraba, que sea compartida con el Norteamérica. Por ahí empecemos, ¿no? Que tengamos si ese sistema que nos interesa, si tenemos una muy buena idea, seguramente lo vamos a poder vender, por así decirlo, en términos de colaboración. And I'm going back and I'm going to say this in English now, that if we have this very interesting question with a species or a group of species that share the distribution with whatever part of North America, I'm sure we can contact any of you or any of numerous researchers in North America and Canada, and certainly we will be able to build a good collaboration. So keep that in mind too.
Yeah, for sure.
Other than just providing tissues.
Yes.
Or than just providing tissues, or sampling permits.
I want to point out on all of those collaborations where the guys come up, I'm your middle author. They're the first author, they're advisors of the last author, and I'm X of it and all. So. I just want to see the stuff done.
We want to add something.
I just wanted to mention that the field of molecular ecology and evolution has always been driven, in my lifetime at least, by technological breakthroughs. There was the Alzheimer era in the 60s and 70s, then came the restriction enzyme era and the cytoplasmic genomes in the 1980s, then came the microsats in the 90s, and now we're into the sequencing generation where next generation sequencing promises to change everything. And with every new technological leap of this sort, of course, people rush in. It's a bandwagon enterprise for a while as more and more labs take up the latest technique, which you need to do typically to publish. It's very hard nowadays to think of publishing an Alzheimer survey, for example, even if it might be entirely appropriate, because reviewers will simply say you're not using the latest technology. But I'd like to do a thought experiment just for the fun of it. Let's suppose that the history of science the history of our field was exactly reversed. Let's suppose that in the 1960s we had next generation sequencing, and then somebody all of a sudden discovered Alzheimer's. There would be a tremendous rush into Alzheimer's work because people would say, wow, this is simple, straightforward, you can read the genotypes right off the gel directly. Furthermore, we're getting to the real stuff of evolution because we're at the protein level, not just all that DNA, which everybody knows is strictly neutral. And then let's take that argument one step further. Let's imagine that all of these molecular biologists suddenly went outdoors and discovered organismal phenotypes. They would go, wow, this is incredible. Look at all these phenotypes that we see in nature. Birds have wings, snakes have no legs. And just think of the world of opportunities that would open. And there'd be a headlong rush into outdoor biology as people would finally find the phenotypic interface with the environment and all the research opportunities that would provide. So I think it's always useful to remember that when you choose a particular molecular technique, it's always best to just match it to the problem at hand. And that so much of this is couched in the context of the history of the development of these tools that one has to bear that in mind as well. Thank you.
Alguien quiere, Eduardo, por favor.
Es una pregunta más particular para John Gunevis. Es acerca de cuando empezó la tecnofilografía, que fue en el 87, y hasta el 2000 que se publicó el libro, si encontraste detractores a tu nueva disciplina y cómo recibió el mundo académico esta nueva disciplina en aquel tiempo.
You got no question. No. He's wondering what happened between you sort of came out with the term phylogeography and then to the publication. Is people detracting you during the period of time? What was the reaction of people to say how you do it?
I'm a poster child for serendipity in science. I very happenstantially got into mitochondrial DNA work in the late 70s, not through any foresight of my own, but simply because one of my colleagues at the University of Georgia was a biochemist and a physiologist who worked on mitochondria, and I started to work in his lab so that I could learn restriction digestion techniques. I never dreamed that it would lead to a field called phylogeography. That word wasn't invented yet, and I hadn't any phylogeographic concepts in mind, believe me, when we started. It was all serendipity. So I think this is a good lesson for those of you that are just starting out in your careers. If you just keep your eyes open, you may stumble on something that will turn into much more than you ever had imagined at the outset. And keep your mind open to that possibility and capitalize on such an opportunity should one or more such opportunities arise in your life.
There was a specific question, if you had any critiques about the field, and how did you deal with it?
If I had criticism.
Criticism. Scientific.
No, again, I would say I was very lucky that I don't feel like I've experienced any pointed criticism. I had some tangential criticism from, for example, from the vicariance biogeographers and so on. But I've always been astounded at how well the field of polygeography has generally been accepted by most of the scientific community and integrated. My own hope is that through my work and my students' work and other collaborators, that we have laid a foundation for polygeography that will withstand the test of time, that will be a solid foundation upon which further structures can be built by upcoming generations of students. But no, I've been largely immune to, or not subject to criticism that I'm aware of. Are you aware of something that I should know about?
Sí, ¿hay alguna otra pregunta? Lalo, está bien la respuesta, supongo. No hay mucha sopa aquí. Por favor, si alguien tiene alguna otra pregunta, un comentario que aportar. ¿Alguien dijo allá atrás? No creo que...
Buenos días, yo quería preguntar. Buenas, disculpe, desde el punto de vista de la filogeografía, ya entendiendo los patrones y los procesos que llevan a la generación de biodiversidad en México, en Norteamérica, desde el punto de vista de la filogeografía, ¿cuál es la mejor estrategia de conservación para permitir que estos procesos para permitir que los procesos que generan la biodiversidad continúen? Bueno, específicamente, desde el punto de vista de la filogeografía, ¿cuál es la mejor estrategia de conservación? Porque los procesos, bueno, conllevan a las especies, pero ciertas veces estos procesos están envueltos, no sé, movimientos geológicos, cambios climáticos, pero ¿cuál sería la mejor estrategia de conservación con los datos reunidos en los patrones y toda esa búsqueda que se hace en la filogeografía?
Bueno, yo voy a comentar, creo que sí, por favor.
The question was that if with the phylogeographical patterns that we know from our work and everybody else's, and the processes involved, if there is one general conservation direction or way of applying strategy, and
Bueno, yo lo primero que quisiera comentar es que, en particular en México, visto desde el punto de vista filogeográfico, nos urge entender realmente los patrones. Y tenemos como ciertos pulsos de entendimiento de cuál ha sido cuáles son los patrones filogeográficos. Pero pienso que nos urge realmente comunicarnos entre los distintos grupos de investigación, no solamente aquí, sino los que vienen de fuera a trabajar aquí, para sentarnos y tratar con la evidencia que tenemos, tratar de detectar cuáles son los patrones más importantes en el país y que eso pueda ayudar a los conservacionistas para poder tomar en cuenta, ¿no?, algunos de estos resultados. Todavía no tenemos una visión en México, y mucho menos continental, de cuál podrían ser los patrones. Yo pienso que ese sería como el primer paso.
Yo les comentaría que, a pesar de lo que yo platiqué de patrones concordantes y de múltiples especies, hay que pensar a dos niveles, porque hay casos que van a tener que ser especies específicos, sobre todo aquellas especies que ya sabemos que tienen problemas de conservación, que a lo mejor ni siquiera podemos ponernos a pensar en la parte filogeográfica e histórica, sino que hay que ser cuestiones más poblacionales e inmediatas, y que por supuesto también está esta otra parte de patrones concordantes que provienen ciertas áreas en las que sabemos que hay gran diversidad, hay patrones que nos están indicando que ha habido evolución, etcétera. Serían áreas que uno tendría que pensar en términos de conservación, más allá de cuestiones de si vamos a poder predecir patrones evolutivos basados en patrones geográficos, creo que no, pero sí podemos pensar en cuestiones aplicadas en regiones de múltiples concordancias geográficas, pero también tiene que ser en el otro extremo específico.
Pues sí, como explicaba la doctora Vázquez, ahora sí están realizando planes de conservación en el estado de California que considere niveles de variación genética en los organismos que están planeando áreas protegidas para proteger el área que contienen la más variedad genética.
Sí, pero menos filográfico que poblacional.
Bueno, los estudios filográficos se informan estas decisiones. Por ejemplo, si vemos que hay un área de contacto entre dos filogrupos, este puede ser una área buena para ser protegida. Y también están intentando en la posibilidad la posibilidad para los animales adaptarse a cambios climáticos. Por eso queremos considerar áreas que tienen mucha variedad genética. ¿Más o menos?
Pero estamos todavía mucho de eso. ¿Alguna otra pregunta, por favor? ¿Quién va a venir a clausurar este evento? Se llama. No viene el director, no viene el secretario.
Tengo una pregunta. Déjame puesto.
Otra pregunta, Alfonso, luego Eduardo.
Bueno, mi pregunta tiene que ver con los planes justamente de conservación. ¿Qué tan comunes son que estos planes consideren esta diversidad genética? ¿Qué tan común es o qué tan, porque me parece que hay como esta brecha entre los manejadores y las personas que se dedican a este campo?
The question is, how do we bridge?
He's asking how often is being used and applied this paleographic and genetic information in conservation programs. I guess California is the leader one.
¿Qué opina usted?
En México, no, como decía Francisco, en la legislación mexicana, por lo menos de la norma oficial de especies, todavía no se considera ningún dato de variación genética. Se están haciendo reclasificaciones de las especies incluidas en la norma y están por lo menos preguntando si se tiene preguntas como tal, si se tiene información de diversidad genética, no está incluido en la legislación, pero ya están al menos preguntándolo. Pero en la formación de áreas naturales protegidas ya empieza a suceder y yo tengo la suerte de tener un ejemplo bueno al respecto porque hemos estado trabajando varios años en la isla de, varios años, hablo de como 15, en la isla de Cozumel, donde tenemos muchas especies endémicas de flora y de fauna, y hace muy pocos años, muchos años llevamos tratando de implementar un área natural, que se desarrollara un área natural protegida en la parte terrestre, porque existía para la parte marina, y hemos generado muchísima información poblacional y genética, y finalmente, hace un par de años, logramos que se nos tomaran cuenta de esa información y parte de la definición de la actual reserva terrestre de la isla de Cozumel incluye la información genética para todas las especies para las que la teníamos. Entonces, también hay otra respuesta. No se hace muy seguido y luego muchas veces no tenemos la información aunque lo pidieran. Pero creo que sí, es un poco de esfuerzo de picar piedra de nosotros, de ir haciendo evidente a quienes crean las cuestiones de conservación, las leyes, que esa es una información sustancial y que ya existe, pues, ¿no? Tenemos poquita, pero por lo menos que tenemos se utiliza. Entonces, para parte de México.
Bueno, según mi punto de vista, es más y más común que los planeadores conservación se consideren niveles de diversidad genética, pero no es tan común que las áreas protegidas que planeaban se realizan. Entonces, hay un...
Recordar, no hay una conexión.
No hay una conexión entre los planes y lo que ocurre en la realidad.
Does New York have any example of using genetic diversity conservation as a practice?
Yes, in the weirdest way. So actually, this is a really cool example. And guess what animal is involved?
Okay, bird.
No, there might be birds. I haven't paid attention to this. But so, for instance, a really cool study on rattlesnakes, right? The timber rattlesnake. They hibernate in these huge dens, okay, in these rocky outcrops in the mountains. And there's major roads that go between these rocky outcrops. And they've done big microsatellite studies to show that there's absolutely no migration anymore between these rocky outcrops, that there was migration in the past, and that there is none anymore, and that the diversity, the pool of diversity is shrinking massively in these rocky outcrops. So the implementation of this is to deal with the roads and move traffic and things like that. In my home state in southern Illinois, they closed roads for the migrations, for the snake migrations. It's one of the only places I know you cannot drive these roads in the fall because the snakes migrate from the fields and forests up to the rocky areas because they know what kind of effect this has. They just did a huge study that a snake crossing the road the size of something like a Pituophis, a large snake, takes over 2 minutes to cross that road. And in that time, these moderately trafficked areas have 70 cars go through. So there's almost no probability that these animals are going to get across the road. And we're seeing man-made effects on genetic diversity, right? So that's a New York example.
Yo me atrevería a decir, Alfonso, que ni siquiera tenemos idea en un gran porcentaje de si genéticamente hablando todos los... Si todos los endémicos a las islas, llámese plantas o animales, son genéticamente diferentes de sus parientes cercanos en los continentes. Son poquitos los ejemplos donde sí hay, bueno, Juan sabrá tal vez un poco más de esto, pero tengo la impresión de que en su mayor parte aún desconocemos si todos los endémicos de los sistemas insulares, por ejemplo, son genéticamente diferentes de sus parientes cercanos. Entonces, el reto es inmenso, mucho menos en sistemas montañosos donde suponemos la distribución es continua y hay un poco, ¿no? You were going to say something, right?
I'm sorry, I missed most of this because no habla español. But I guess the question I think was, can we give examples of conservation applications for phylogeography? I would like to give a hypothetical possibility that's just a dream of mine, but it involves the notion of what I call phylogeographic parks, or Pleistocene parks. And the basic idea is that, think of the United States right now, for example, or indeed around the world, there are many national parks that are set aside typically to preserve unique geological features of the environment. So, for example, we have Yellowstone National Park with the geysers. We have Glacier National Park with the glaciers in the high mountains. We have Yosemite National Park with the magnificent waterfalls and so on. This was a landmark conservation initiative when national parks were first established in the early 1900s, late 1800s. I can imagine a time where an enlightened North American society would designate what I'll call phylogeographic parks. These would be biodiversity parks, places where there were centers of endemism or unique regions that are particularly rich in a historical biological sense. Imagine setting up parks around such a notion with red riddles, for example, regions, ecoregions in the southwestern United States. There would be one phylogeographic park in northern Baja, another one in the Sonoran Desert, and so on. And with each of these, there could be interpretive centers, so the general public could come and see the history of this area and what makes it unique and why organisms occur there and how they differ from organisms that occur elsewhere. So it would be extending the national park concept now to a biotic arena. So this is just a dream of mine, but it's an example of how this could play out in a conservation context.
I think you would have to turn television off for a generation, eliminate most sports, and then maybe you'd be smart enough as a public. And the iPhones, yes, exactly. And then we might get there.
Sí, claro.
La filografía nació como una disciplina muy simple, es decir, sobreponer el patrón filogeográfico en un mapa y sobre ese patrón tratar de explicar los procesos. Después se generó el análisis de clados sanguíneos, que hace unos pocos años le dieron el golpe de gracia, aparentemente, aunque siguen publicándose artículos con clados sanguíneos en 2014. Ahora parece que hay una tendencia hacia una filografía más matemática, más de modelos. Es decir, cómo vislumbran el campo de la filogeografía en los siguientes años.
Claro is saying that paleogeography was born in a very way to overlap biology on the geography of the distribution and with that try to describe the patterns. It then started developing and we had nested trade analysis, which was probably one of the first more thorough hypothesis testing sort of way, and from that it has still growing and growing into much more methodological and analytical discipline. And the specific question is, how do you envision phylogeography in the future in terms of it's going to be only extreme analysis, analytical exercises, or what?
This is my favorite question. So the future of phylogeography is the past of human population genetics. In almost every case, what human population geneticists were doing 5 years ago is the stuff that we're really getting excited about now. So we saw this with approximate Bayesian computation, for example. In the early 2000s, there were people like Jonathan Pritchard who were building these really nice models of human demography and testing them using ABC. And then 5 or 6 years later, people who were interested in phylogeography started doing that. We saw it with things like diffusion approximation approaches for using allele frequency data to estimate demographic history. And so I always tell my grad students, if you guys want to cutting edge in phylogeography, just read human genetics papers that are 5 years old. And that's the way to go. That's our secret.
I feel a little weird talking about the future of phylogeography in front of John Hughes.
Like music, we were talking about, clouds.
Yeah, it's like my telling the Rolling Stones. So, in my opinion, I actually want to hear what John has to say about this. Phylogeography has increasingly recognized itself as a discipline that is answering population genetic questions. And like any scientific discipline, as the theory has developed, it's become necessarily more analytical. And so I suspect that, yes, the future of phylogeography will be one that's very much dominated by models of sequence evolution and population genetic models and coalescent models. But there's a good reason for that, which is that we're recognizing only now really how to analyze population genetic patterns in a spatial context.
I don't disagree with anything that was just said, but I might add also that another direction that I think I can already see phylogeography going, and has been going for several years, is when I travel around the world now to such conferences, it never ceases to amaze me how many people are doing phylogeography on new taxa and in new biological settings. And if you think about it, the percentage of the biological world that's been explored from a phylogeographic perspective is absolutely minuscule. We've barely begun. So I can see the kind of studies that Brett Riddle was doing in the southwestern United States, or that we did in my lab in the southeastern United States. These kind of studies are now being conducted around the world. I've seen it in South Africa, I've seen it in Australia, I've certainly seen it in Europe. So I think there are tremendous opportunities for expanding the taxa and the biogeographic regions that are being explored, all in a historical context.
I got a question for you, John. So when you watch these two guys, all the phylogeography you showed of Central America and Mexico, do you sit there, John, and look, this is all me. This is all my stuff. You take ownership of all of this.
I don't take ownership, that belongs to the people that did the work.
Okay.
But I'm very pleased and proud, I suppose you could say, to see such studies being extended to new biotas. When I see this beautiful work by Ella on Central America, or by Juan on northern Mexico, to me this is very reminiscent of our early days of exploration of the United States and the eastern United States. So I love to see it. I love to see these concordant patterns emerging, which still I find absolutely fascinating, that there can be these repeatable patterns in nature, along with all the idiosyncrasies.
Yo quisiera, antes de las dos otras preguntas, agregar también que me parece que, aunque ciertamente la feligrafía, como cualquier otra disciplina en su desarrollo, incorpora elementos de tipo analítico, y eso tiene que ver, obviamente, en cómo va madurando la disciplina. Pero también me parece que va a llegar un punto en el que vamos a volver a voltear a la biología de los organismos. Y entonces eso puede detonar una nueva revolución. Es decir, cuando descubramos cómo incorporar otra vez la conducta de los animales, la variación fenotípica de los animales, no solamente desde el punto de vista molecular y espacial, sino biológico. Cuando realmente integremos todo eso como en una multidimensión, ya pensando en el futuro, creo que vamos a tener que regresar a observar a los bichos y luego eso tratar de incorporarlo analíticamente.
Yo también nada más añadiría una cosa que salió en una de las diapositivas creo que fue del doctor Rick hoy, en su plática de hoy, que en realidad para hacer esa... Hay dos aspectos de estudios bibliográficos, los que ya tenemos muy buenos ejemplos, donde podemos escribir estos patrones tan a detalle, tan fino, como las especies y los biomas que todavía tenemos que trabajar. Eso es muy emocionante, todavía tenemos mucho que hacer. Pero en estos patrones que ya tenemos conocidos, que ya sabemos qué está pasando con ciertas especies, tenemos que dar, como dice Francisco, ese salto bajarnos en la escala, aspectos ecológicos, biológicos, de historia de vida muy finos, y a mí ahí me gusta mucho la conexión que ya está sucediendo con la genética del paisaje. Y porque es una manera de que nos permite retomar aspectos ecológicos, ambientales, biológicos, pero además asociarlos analítica, matemáticamente, con la genética, que de todas maneras estamos evaluando. Entonces, de verdad creo que ahora la genética del paisaje, que ya estamos hablando de 10 años de formación del término genética del paisaje por Stephanie Mann, en realidad pues nos va a permitir otro salto.
Tenemos aquí dos preguntas, por favor.
My question is related with this and it's about all of you work with the reconstruction of speciation patterns or relationship between the species. Some of you have worked in question about how we can reconstruct interaction between species or something like that. That I think that maybe if we can reconstruct the interaction between species, we can explain other things about these patterns, and I think that maybe it could be a new topic in the phylogeography.
Do you want to address that?
I think so. So
Yeah, is your question about how interactions between species give rise to speciation, or is your question about how biotic interactions may shape phylogeography? The second one.
Reconstruct interactions between.
In the analysis.
Okay, I'm not sure that my answer is going to be very satisfactory, but for example, today I showed you those niche models in which we reconstructed the paleohistory of joshua trees, Abiyakas. All of that reconstruction is considering only abiotic factors, and that's a problem. So it's only considering climate or soils, and that's a problem in two ways. Firstly, it is quite likely that ecological interactions have shaped the actual distribution, or that is, there's a difference between the fundamental niche and the realized niche. But secondly, our actual estimation of the niche using that presence data, so we looked at where joshua trees occur now, and then tried to surmise their potential distribution. Where they occur now is also limited by biotic interactions. And so, at least in the ecological niche modeling context, we need some other way to try to incorporate biotic information. And I don't know, we don't have bioclim for species distributions and interactions. In a macroevolutionary context, so thinking across phylogenetic trees, people are trying to work on those things, either using phylogenetic state reconstruction or looking at how changes in ecological associations affect diversification rates. On a phylogeographic scale, I think we're still not there, and I think that's a big piece that's missing.
So with respect to the level of what I was doing with the interactions and what feedback you would expect on phylogenies, the high-end programming for that, I have 2 points to make here, is just beginning. Okay, some of these things are only a few years old, and there's more than I showed you. And then, and there's one coming out within this month by Luke Harmon, which is the impact of species interactions on phylogenetic trees. And he has some massive new method. He only, like a smart ass, he only showed me the title and wouldn't even let me see the abstract. I don't know if he's trying to build up some site for this. Yeah, exactly, and it's going to be amazing. So it's in its infancy. Being that it's in its infancy is really good, and it's really good for you guys if you're interested in these questions. And they almost all exist entirely in the R framework. So the second part that I was going to say is, become good statisticians / mathematicians, learn R. You will be able to learn statistics and the mathematics associated with all of these things in R very easily. It's going to be a little bit of an uphill climb, but once you get it and once you learn it, and I'm assuming some people don't know it, maybe most of you guys do, but it's addictive. And once you can get it and you can work, it's better than sitting around seeing what some dummy said on Facebook, right? It's so great. And sometimes when I write my code and I figured it out, and all I have is one last line to write, I'm so happy. I'll go and get a beer or a big coffee and I'll relax, and then I'll do the last piece, and then it's beautiful and it's done. And you have these weird patterns like I do. I go to sleep, I can't figure it out, and then as soon as I wake up, boom, I figured out the answer. So you can do so much, and you can add so much sort of to this world once you learn this. You're not a slave to whatever somebody else can make. You can make your stuff. If you have the question, you have the idea, you can make it. And it's not that hard. I would say take it super proficient in it anywhere between 3 months and a year if you're really working on it. And it's just a language you can speak. Then you can look under the hood. If somebody comes up with a new package, you can look at all their code. You can see how it's written, remodify it for your purposes. And so there's a lot of questions that we couldn't ask before. And there'll be a lot of questions that you can't ask if you're reliant on other people making stuff for you. So while maybe it's going to be difficult for the next gen world to arrive in Mexico like it has in the US, you can be prepared with R for sure, and really be good at it. So I'm very excited about this as a way to open up your brain, and you will really learn stats by learning this.
I just want to second that. So the two software packages that I talked about yesterday are both almost entirely written in R, and they wouldn't be possible without all of the other functions and packages that we have used that other people have developed. And because this is all open source and it's all freely distributed, it's the kind of thing you can pick and choose, and you can go shopping in the smorgasbord. I'm going to use Brunel and Yang's function for calculating Bayesian probabilities, and I'm going to use Cummings' function for calculating GSI. And you put it together and you build something new. And so it's like a big box of Legos that we all share, and it's free, and you can do really creative things with it. And it's really it's a great thing for the field. And so I would second that. If you don't know R already and you're interested in any of these things, invest the time because it'll pay off.
And there are tons of tutorials, even in the Philo realm, tutorials that will teach you how to get proficient in it. My feeling is the best way to get really good at it is have a question and a potential problem and work through those routines, and then you'll discover something about your data. Instead of doing it blind,
delete Microsoft itself from your computer.
Yeah.
And do all that.
And I wanted to also mention there's this other programming language that's just out. It's called Julian, and it is unbelievable. All the big programmers are using it. It's as simple as R, but it compiles on the fly, and it's close to C++ fast. It's extremely fast. The libraries aren't all built yet, but the predictions from the computer scientists that work in phylogenetics are saying, in phylogeography, are saying 2 years. And it'll be up. And it may eclipse R. It's unbelievable. I learned it in 10 minutes. The tutorial is so unbelievably fast. It is, you know how Macintosh is intuitive when you use Mac? This programming language is intuitive like that. If you know R, yeah. But, sorry, if you know programming, right? But it's okay. It's okay.
La otra pregunta, por favor.
Sí, I have a very complicated question because from your point of view of what happens when you are trying to save a species that is already extinct in the nature, like frogs. I'm really involved with frogs, and this massive extinction that has been provoked by this awful kitri family. And I want to know if there is really a hope to reintroduce the species to the nature when you only have a few number of individuals, or what happens with the genetics of the population? Is there a future really, or is not going to be a fact that it will be saved the species?
You're saying if you're only raising them in zoos, they have no habitat or place to go, what do we do?
I'm trying to say that what happens when you only have a limited number of individuals to repopulate or to make a new population and introduce them to the nature in the wild.
Yeah. So part of your question is like, can we ever save these species? And I don't know the answer to that question. That's, you need to ask the curadera to think of the future for now. But I can say 2 ways that phylogeography can help with that problem. One is using information about genetic diversity to design managed breeding programs in such a way that you reduce the loss of genetic diversity. So in very, very small populations, we know that inbreeding can be a major factor that ends up being the coup d'etat in knocking a species out. And we've seen, for example, in Florida panthers, or in the lesser prairie chicken, or in snakes in Sweden, that inbreeding has been a major factor in reducing species. So if you can use information about genetic diversity, either proxy, because you have samples of frogs that are taken from different populations, or you assess it directly using microsatellites, for example. You can try to plan breeding in such a way that it maintains genetic diversity. The second area where I see phylogeography can potentially help with this is if you suspect that your species once had a much more widespread range and you want to plan what might be potentially acceptable places for reintroduction, you could use the ecological niche modeling approaches, taking known historical records to try to predict what are potential areas where the species can occur. Does that make sense? Thank you.
Y si tienes la suerte todavía que además tienes los datos filogeográficos de alguna de las especies de ranas o varias que también, bueno, por ejemplo, en la Baja California hay información filogeográfica de varias de las especies de ranas, que además es una de las zonas especies más atacadas por el virus, bacteria, hongo. Puedes también además considerar estas zonas de diferenciación, que aunque los microsatélites a lo mejor no te revelen que hay diferencias, tú sabes filogeográficamente que tienes linajes distintos, también es una guía para ver de dónde tú podrías tomar individuos y a dónde llevarlos y a dónde no llevarlos.
¿Alguien más?
I wanted to give one negative point of view to this because I mean, just from, I don't need this microphone. Can I just talk like a normal?
Okay. Okay. My feeling is, you know, the elephant in the room is overpopulation, and we do not see this stopping. There's not even a political discussion anywhere about controlling human population. So wherever I go, whether it's the most remote part of the planet, remote part of Madagascar, where people haven't surveyed in 100 years, there's humans in there with cattle. There's always something. So you might be able to help one or two species at a time, but I think stemming the tide of human beings is the real issue, right? I mean, you just don't see them stopping. So in your lifetime, I don't know what to say. Get used to seeing a lot of extinctions, unfortunately. I just don't think, and we don't have the resource to do all of this, right? I mean, that's another aspect. And I know that's very negative to say, but if we're never going to have a discussion about overpopulation, we're still on exponential growth, and we haven't turned that exponential growth into logistic growth, which is going to be horrific to see probably how that happens. I don't see the hope for most animals. For instance, go to Brazil. We think of Brazil as wonderful because they have all the world's tree frogs and biodiversity of frogs. This is common in Brazil. Most people hate frogs. It's a common thing to hate frogs, okay? It's a very weird thing. People do not like frogs. So, outside of the scientific realm. So, I just don't see it happening. I think that George Carlin is this American comedian's point of view, which is just like, oh, we worry about the Earth isn't going anywhere, humans are. So as long as we have a few lineages left after all the humans are gone, they'll diversify and fill all these wonderful niches and everything else. So I guess that's the only silver lining on my whole negative statement is when the humans are gone, things will recover. I don't have any other positive view of that because I don't see chipping away at one species at a time or two species at a time is going to make an overall difference on massive crises that we're sitting in. So, sorry.
Okay, now to prayers. Thank you. Eugenie.
Tengo una pregunta que es un poco existencial y que también le quería hacer al doctor Rigo, pero he visto que no puede estar aquí. Sí, le toca a él.
Le mandamos el correo electrónico.
Bueno, tengo que regresar un poco a la discusión del principio, pero si bien la biogeografía y la filogeografía buscan entender o encontrar patrones geográficos, a veces me preocupa un poco cuando me llama la atención saber por qué siempre elegimos grupos de especies que creemos que responden a una barrera geográfica y que cuando no sale que tienen estructura no buscamos una explicación diferente, sino siempre no tuvo estructura y el grupo que sí tuvo una respuesta, no sé, alergia, ritmo, bueno, estoy hablando de patrones en México, y eso me hace pensar que por eso no podemos descubrir muchas de esas especies crípticas de las que hablaba el doctor Riddle y también muchos de esos patrones que nos ayudarían a entender a los otros grupos que no tienen esa distribución. Digo, es más fácil quizás echarle la culpa a una barrera si es que realmente corresponde con ello, pero también es difícil que podamos ver otros patrones si no estudiamos otras especies. E incluso muchos de los trabajos filogeográficos actuales no incluyen a las especies hermanas. Chichi, vas a tener que resumir porque no lo puedo traducir. Ya me perdí de la primera parte. Sí, es eso. ¿Por qué estudiar? Perdón. ¿Por qué no estudiar o qué piensan acerca de por qué no estudiar otros grupos que no respondan? O sea, porque siempre estudiamos grupos que responden a barreras y siempre explicamos si responden o no a esas barreras, pero no podemos explicar otras, la distribución de otras especies que no responden a ellas.
I'm going to translate it literally and then I will give my point of view. She's asking why we don't study species that do not show a strong structure and phylogeographic patterns, and with doing that we're missing information from those species that do not show the patterns. And I'm going to answer this in English, so I don't have to repeat myself. I don't quite agree with you. I think we do study whatever species we're interested in, and if they show a certain pattern, we try to explore what are the processes beneath them. But we also have those species that do not show a pattern, and we also try to explore why. So I still don't understand the question, or I don't quite agree, but that's what I think. Jen?
I think I got the gist of the question. I would say, I think if you review the literature, which we have done, you will find that many species that lack phylogeographic structure have indeed been studied. And in fact, about 50% of all species that have been studied lack substantial phylogeographic structure. In other words, they are composed of just one, what we might call phylogroup. Another 30 or 40% have 2 to 3 or 4 phylogroups per species. One of the things that's always amazed me is that the number of phylogroups in the biological world shows this good of a correspondence as it does with the number of recognized biological species, despite the fact that they are very different criteria. It leads me to believe that phylogroups have some real biological meaning.
So, while I haven't done the kind of surveys that John has done to really get numbers on it, I think that it does seem to be the case that it's unusual to read phylogeographic papers and hear the authors say there isn't really a pattern here, there's not something that we're getting out of it. And I think that in some ways this supports one of the points I tried to make yesterday about how we want to read a lot into our data. And once we go through all the hard work of collecting samples and collecting data and analyzing the data, it's often very hard to say, well, guess what? We can't tell you anything. There was nothing. And so we have this tendency to elaborate as much as we can. And it's often hard to sort of know what people mean by a pattern that's appropriate or significant or substantial, or what phylogeographic structure is, because these are terms that are defined in a number of different ways in the literature. And lots of people use terms that were originally defined very well and very rigidly. And over time, the meaning of these terms expands and loosens and changes in ways. And if you read a paper and they say, phylogroup, and you're thinking about it in the context that it was originally defined, and they might. One of the things that, as reviewers of papers, that we always need to do is really force the authors to be precise with language, so that when someone says phylogroup, they're either saying it in the original meaning of the term, or they're at least defining how they're using the term.
Oh, I got a point about, a lot of our
stuff, a lot of our stuff, like for instance with the boas that we did, and you find 2 big deep groups, they're probably 2, and a lot of the stuff probably 2 species. So then what you've done is found each one with spectacularly no phylogeographic structure from 2 species, if that makes sense, right? So they do happen. And I'm working on these snakes in Florida right now that are so spectacularly uninteresting. It's amazing. I mean, spectacularly uninteresting. Yes, exactly.
Nada más quisiera preguntar a nuestro moderador de tiempo hasta cuánto tenemos rentado el auditorio. ¿Ya nos abandonó? ¿Ya se durmió? ¿O podemos seguir aquí la discusión? ¿Cuál es cuál es nuestro programa? Se agota mucho, créenos, no hay
bueno, ahora vamos a pasar a la entrega de las constancias. Queremos agradecerles por acompañarnos en este coloquio. We are going to give the certificates for the student colloquium. We are very glad. And well, for John Davis. Now we want to thank for all the staff who was helping us, and we want to deliver the certificates for. There were so many people who was helping to run this event, so we want to deliver their certificates too. For Carolina Reño is here. Yeah, please. For Jimmy, please, come on. For Cristian Luis Rodriguez. Thank you. For Carlos Antonio Bella, who was working all this time. We are YouTube, please. Say, like us on YouTube, please. Remember. For Michelle. Where is Michelle? Michelle is. Michelle. Well, Michelle is out. For Pablo was working very hard on the photography contest. Michelle is gone now. Thank you so much. And for Lorelli, Lorelli was here. Yeah, come on, Lorelli. What? Hector, I believe Hector is on the gas line.
No entienden su nombre, Alfonso.
Okay. Para